Well, good morning, everyone, and welcome to a cold, blustery November day in Holland, Michigan. Kristen Rankema is an assistant professor
Let's
in the
try it again. Start
Biomedical
over.
Sciences Department at Grand Valley State University. She received a BA in
True.
biology and writing at
Have been
Anderson
told this.
University and completed her PhD in immunobiology at the University of Arizona. She also did postdoc work at the University of Minnesota. Kristen is a native of West Michigan. Her lab at GVSU is interested in how microbial exposure impacts immunity. They hypothesize that the human immune system needs proper education, which comes from immune responses to viruses, bacteria, etc., in order to develop properly. If that education doesn't happen properly, then they suspect there is an increased risk for potential long-term consequences, such as asthma and allergies. Where Kristen's lab differs from other labs is that they are interested in whether microbial exposure impacts immune responses to cancer. They are currently studying this using a mouse model of melanoma. Okay? And so without further ado, I'll turn it over to Kristen Rankema.
Sorry? Oh, yes. I could have done that too.
Yeah. Why don't you go ahead and, because I don't want to mess anything.
Yeah, no, you're good. Where's our...
There
Here
we
we
go.
are. All right.
Oh, no.
We got it. Never worn a microphone quite like this one, but I think we're good to go. Awesome. Thank you very much, Kim, for the introduction. I can tell you that these past couple years have really thrust me into teaching in ways that I never thought I would. And so I'm pretty used to having to teach simultaneously via Zoom and also in person. I'm also used to having to record things, so hopefully this will go relatively smoothly. And so today I am going to focus on something that used to be a huge part of my research career, and that is studying how aging influences the immune system. And so this was actually the entire focus of my dissertation during my PhD at the University of Arizona. And the University of Arizona has one of the leading centers on aging research. And so it was a real great opportunity for me to be involved with that research there. So I'll touch a little bit on, that type of research too. So I'm going to start with what I like to remind all of my audiences that I ever have, whether it be an undergrad, or, all the way to HASP, whether it's professional audiences or non-professional audiences. And that's to put in the context of the immune system, what are the major functions? Why do we want our immune systems to function properly? And so, in my opinion, there are some major goals of the immune system. And the first one is to be able to very quickly and accurately recognize something that is dangerous, right? And so we really need to be able to respond rapidly and effectively to certain what we call pathogens. And a pathogen is a very broad term for things that mean basically things that are capable of causing disease. So these are things like viruses and bacteria, certain fungi, parasites, things that can cause disease in humans. So pathogen, you might hear me use that term. I slip into that term use a lot. What I mean by that is something that can cause disease. So we want our immune systems to be able to recognize that something dangerous potentially has entered and there needs to be a response. Obviously, we don't stop at the recognition of something dangerous. We also want to deal with it appropriately. So we're hoping that our immune system can both kind of contain whatever that disease-causing organism is, as well as ideally eliminating it completely from the body. Now, that doesn't always happen super effectively. You may be aware of certain types of viruses, especially, that tend to stick around in our bodies for a long time, if not permanently. So for example, the chickenpox virus will actually live in certain neurons and the nerve endings, and for many people, never ever cause a problem, even though it's still there. However, in other individuals, unfortunately, that virus will reactivate and make shingles. So sometimes the immune system actually can't get rid of whatever the pathogen is. But ideally, if we could have a perfect world, our immune systems would get rid of it completely. Now, this is also a major goal that I think some of my colleagues kind of forget to mention. So imagine that you have a bacterial infection and your immune system recognizes it, responds to it, and eliminates it. Do you think it would be a good idea to keep a very active, robust immune system going when that bacteria is no longer there? You can probably guess that no, that's probably not a good idea, right? So it's actually a very important part of the immune system to turn off when it's gone. And we'll talk a little bit more about that too. And then finally, I'll also touch on this. It is also a major goal of the immune response to make what we call as immunologists immune memory, okay? So this is a hallmark of certain cell types in the immune system that actually can stick around after the pathogen has been taken care of and protect you long term against reinfection, okay? So we'll also talk about immune memory, the goal of vaccination in making immune memory, and what we know regarding aging also for that. So we'll kind of touch on a lot of these topics, and I'll kind of let you know where the field is at in terms of understanding what happens with aging. So I can tell you that these are the goals of the immune system for every human being on Earth. And so it is also shared in the aging population. And generally, immunologists, when we refer to the aging immune system, we kind of picked a random age, I feel like, and we say 65 years and older. So in general, if I share data with you that I'm saying, these are the findings in an aging population, generally speaking, that's the cutoff, okay? Just in terms of how ages are grouped by many research labs. So the goals here are still the same for aging population, but unfortunately, we know that things don't always go as planned. So we'll talk a little bit about what happens in the immune system with age. So first of all, some background about why we care about this. I probably don't have to convince this audience, but sometimes I have to convince my undergrads that this is a worthy cause. So why do we care, right? Currently, 1 in 8 people in the United States is age 65 years and older, and that is only continuing to grow. It's estimated that by the year 2050, this will reach 80 million people. So people are living longer, and because of that reason, we really want to be able to keep people healthy and happy as long as possible, right? And so we actually use a term in the field. So we talk about lifespan, right? Lifespan's just increasing life expectancy. What we're really interested as immunologists is something called health span. I'm not sure if you've heard that term before, but really what that means is we don't want to just let people live longer, we want to have them be healthy. So health span is a term that's becoming more familiar, especially in immunology, because we want to make sure that in addition to maybe increasing life expectancy, that people can be healthy at the same time. So what we do know currently is that it's interesting because, aging is definitely associated with a decrease in immune function overall. If I could give a blanket statement, that's what I would be able to say. And we know that that decreased overall function is definitely associated with both increased susceptibility to infectious disease, so you're more likely to become infected, in addition to increased severity to the disease. It's kind of a double whammy, right? You're more susceptible to infection, but you're also potentially more susceptible to the severity or even death due to the infection. And worldwide, infections are still some of the leading causes of death. So this is definitely a very important field, and many of my colleagues agree with me there. So what's really interesting and why there are quite a few researchers that study specifically the immune system with age is because the immune system seems in particular to be very sensitive to aging. So just on a very basic kind of cellular level, we know that cells in your body, all cells, can be impacted by age. They don't proliferate or replicate very much as compared to younger cells. There's some very general kind of aging hallmarks in your body. But what's weird and still not completely understood is immune systems seem to go beyond that. The immune cells seem to be even more susceptible to some of those aging hallmarks we see. So we think it's really important that not only study aging as a whole, but also really focus on the immune system in particular. So I can tell you that as a good thing, in my opinion, and more as I kind of follow the field, specifically studying aging and immunity is definitely a top priority of both scientific research, like what my lab does, the basic science, as well as then translating that to actual healthcare. And we use an expression in research, we say from bench to bedside. You may have heard that expression, but we start at the bench really trying to understand the basics, but it's always our goal then to translate those findings to actual benefits at the bedside in healthcare. Okay? So I actually just got back late last night from an immunology conference in Chicago, and I was actually surprised and pleased to see how many presentations touched or even completely focused on the question of aging and immunity. So I can definitely share with you that, this is a growing field. All right, so I'm going to go over some basics of how the immune system works, and then we'll go in a little bit more detail. I kind of pick and chose some more specific examples of what happens to particular components of the immune system with age. Okay? So if you've come to a talk with me before, some of these things will look a little familiar, which I don't think is a bad thing. So you may recognize some of these upcoming slides. So if we consider what happens during a normal immune response, we have a person who gets infected with a pathogen. And most commonly, when we study pathogen, which remember is something that can cause disease, we're usually looking at either bacteria or viruses. Both bacteria and viruses are considered microbes, meaning they're teeny, teeny, tiny. You can't see them with your visual eye. You have to use microscopy to see them. And bacteria and viruses can cause quite similar diseases, but the major difference between bacteria and viruses, at least right now, is that we would consider bacteria to be alive. They can replicate by themselves. They have the hallmarks that we learn in like general bio of being alive. Viruses, on the other hand, are different. Okay? So they still cause disease, but they're very dependent on you to cause that disease. So viruses actually cannot make more copies of themselves unless they get inside of your cells to do it. They don't have their own machinery to be able to replicate or make more of themselves. So bacteria don't need our cells to do that, but viruses do need our cells. So that's the major difference between viruses and bacteria. I'm not really going to go too much into that, now. Right now, just consider you get infected with something, okay? So let's kind of follow what's going to happen here in the body. And the way that I depict this is in a graph form where we'll have the number of either the pathogen or the immune cells responding, and you'll see that in a second, over time. So what's happening over time during an immune response? So let's say you get infected, unfortunately, with a virus or a bacteria. The first major goal of that pathogen is to make more of itself, okay? So what this graph is depicting in the red are the number of pathogens over time, and they're increasing in your body. It might be in your nose if it's the cold virus. It might be in your intestines if it's a bacterial bug that gets there. It can happen in, various places, but it's definitely trying to make more of itself. Luckily for us, that sends out certain danger signals. So remember how I told you the first goal of the immune system is to be able to recognize when something is wrong, okay? And there's various ways that our virus or a bacteria sends signals to the immune response, kind of like red flags, Something's wrong here and we need a response. And so what happens is you have an army of immune cells. And the immune cells are your white blood cells, okay? So like when you hear the term white blood cells, you could basically interchange that with immune cells. So these are the white blood cells, and there are many different types of immune cells that we would classify as white blood cells. And I like to think of the immune system as a mobile organ system. It's highly specialized, just like your liver. It has a goal, but the liver is kind of all contained, right? And it does its function in this solid organ. The way the immune system works is it has very defined functions, but it can go anywhere, okay? So that's what I think makes it so special in a lot of ways and makes it very difficult but very interesting to study. So imagine now your white blood cells have identified the problem, and they are going to rise to the occasion. So because of these danger signals that the pathogen is sending, your immune system does respond. And so it's also going to start to build an army to hopefully fight against this pathogen. Now there is a slight delay because it takes a little bit of time, for the immune system to be able to recognize this danger. But ultimately, you want it to, as quickly as possible, respond to the threat. So what we're going to consider first is something that's called the innate immune system. The innate immune system, basically what that means is it's the first to respond to an infection. It's first on the scene. It can very rapidly respond. And its major goal is to hopefully contain and eliminate the threat before you even need to call in the big guns. And we'll talk about what those big guns are in a minute, okay? So this is where we're at in an immune response, early on after infection. So I'm actually going to talk to you about a few of my favorite specific white blood cells, right? So we've got this umbrella term, white blood cells, but what different types of white blood cells are there? What do they do? And what do we know about changes that happen with aging in these cell types? I'm a cellular immunologist, so I really like to break it down into these different immune cell types because they really do have distinct functions, but ultimately have to work together really in this complicated, I like to think of it as like an orchestra piece, really, in order to have the best results. So the first cell type I'm going to mention that really is the first on the scene is the macrophage, which you may have heard of before. And these macrophages are spectacular cells. They line all of the surfaces and barriers of your body that might come into contact with open air or food. So for instance, you've got macrophages lining all underneath your skin. So if you get a cut and bacteria gets in, they can respond within minutes. You also have macrophages that line all your mucosal membranes. So you can think mucosa really is anything in the respiratory tract, your nose, your esophagus, your lungs. You also have that in your stomach and your gut. You have this, what we call mucosa, and you've got macrophages right underneath that too, ready to respond if you happen to get an infection via that route. So these are the first to respond. This is an actual picture of a macrophage, and what it's doing, so this is the macrophage here. It's kind of on a flat surface. And what it's doing is it's actually extending what we call a pseudopod. It's kind of like a little foot or a little hand. And it's reaching for this little bacteria here. And if this was a video, what you would see is you would actually see this macrophage grab that bacteria, pull it in, and do something called phagocytosis, okay? So phagocytosis is a fancy term that means eating. The cell, this macrophage, can grab bacteria, pull it inside, and destroy them. That is its major function. And so because of that, when I like to think of macrophages, I think of them as the garbage collectors in the cell, in the body, right? So they're constantly going around and patrolling. They not only help with cleaning up bacteria, but they also can just clean up normal cellular waste products, or dead and dying cells just happen as normal wear and tear of the body. So they're really important not only in infectious disease. So these are going around and phagocytosing. So just as a reminder, these guys can actually take in bacteria especially and destroy them. And so they're very, very important. I actually have a little video that we're going to try to get to work that shows phagocytosis actually happening. So let's see. We might have an ad because it's YouTube. So just bear with me. My students always think it's so funny when I do this. Let's see if I can get this to work. All right. Yep, of course we've got an ad. Sorry, Zoomers, if you can hear the sound. I'll skip it as soon as I can. All right, so I'm going to pause it for a second. I hope everyone can see this. I know you guys can. I'm hoping the Zoom people can as well. So what we have here is all of these little tiny dark things you see. Those are all bacteria, okay? So bacteria are very, very small, much smaller than the cells in our body. And what you're going to see here is this is our macrophage. So I want you to keep an eye on what this macrophage is going to do. You're going to see it start creeping up. Keep an eye on what it does to this cluster, this clump of bacteria. So here it comes. It's moving its way. These are mobile cells. Now see how it's starting to engage here with the cells? Watch this. It's taking them in. I don't know if you can see these little bacteria cells coming into this macrophage, and they just dissolve, right? So what this macrophage is actually doing right here in real time, and it is slightly sped up, but you see, it can not just do one bacteria at a time, it can just take all these bacteria and they're all destroyed, okay? So I just think macrophages are the coolest. And we do study them in my lab because they're actually highly involved in cancer as well. So we kind of take a different approach, but we're really interested in macrophage function in cancer. So I hope you can appreciate, that these cells are pretty important, especially at containing potentially an infectious agent really early on after you get infected. All right, so we're going to go back to this. So I think we're good here. So what do we know about macrophages and aging? I do have to put a disclaimer here. So it's interesting because even though the innate immune system is the first on the scene, we actually know very little about it. We know much more about the big guns that we'll talk about in a minute than we do about just generally speaking how macrophages work, but then even less is known about how they're impacted with aging. So I'll kind of share with you what we think in the field, but this is certainly something that needs a lot more research. So what do we know about macrophages and aging? And just to remind you, right, the major function of the macrophage is to be able to eat and destroy bacteria, okay? So what do we know with aging? Unfortunately, macrophages with age have reduced phagocytosis. So they have a reduced capacity to efficiently both eat and destroy bacteria. So they lose that function. Not completely, but they are less efficient at it, okay? We also know that macrophages produce reduced inflammation. Now, I haven't touched on that yet, but early on in immune responses, inflammation, which you would probably think of like swelling, right? If I said the word inflammation, you might think like swelling, redness, something that happens like that. So inflammation actually goes beyond that. Inflammation is really a way that the immune system communicates. So this inflammation is a way to really heighten the red flags of an infection and bring in more of an immune response and get things really going. So macrophages with age have a reduced capacity to kind of send out that warning signal to the rest of the immune system, calling for reinforcements. We also know that macrophages are involved with regular wound healing processes. So if like you get a cut and it clots, right, and then it's eventually over time, you get a scab and it starts to heal. We now know that macrophages are actually really important to that process. And so not only does this reduced macrophage function impact reduced immune responses with age, but we also think it might play a role in the delayed healing process as well for certain types of injuries. Very little is kind of really defined, but there are some researchers who are studying that as well. So these are some of the major things that we think we know currently about macrophages with age. And so ultimately what this results in is these defects that happen with age in macrophages do contribute to that overall reduced response to infection. That is kind of a hallmark of the aging immune system. So these are the macrophages. Very important, very cool, and are definitely impacted with age. So now I'd like to move on to my other favorite immune cell in particular that I want to touch on. Also part of the innate immune system, so still is going to respond very, very rapidly upon infection. This cell is called a neutrophil. Neutrophils are by far the most abundant white blood cell in your body. They make up about 70% of your white blood cells, okay? So these neutrophils, I consider them the foot soldiers. They are very good at rapidly responding to an infection, and they're very short-lived. The average neutrophil only lives about 3 or 4 days. And so what their major goal is to like get to the infection, and then they basically kamikaze themselves to try to fight the infection. They're gonna die anyway. Might as well take out some bacteria, while you do it. So the way that they do this is through 2 major mechanisms. The first one, I think, is probably one of the coolest mechanisms in all of immunology. And I don't even study it. I probably should. So this is called netosis, N-E-T. So they're called nets, which is kind of nice because that's actually how they function. But NET is actually an acronym. It stands for Neutrophil Extracellular Trap. And so what it is this neutrophil, here's an actual picture of a neutrophil here, and then a net. What this neutrophil actually does is it recognizes that there's an infection, and the best thing that it thinks it can do, if cells could think, would be, I just gotta sacrifice myself to get rid of this. So what it does is it actually spews out its own DNA, which is very, very sticky. And it creates this big net which can catch bacteria. So this is very sticky. So the bacteria get caught in this. And then in addition to being really sticky, there are like these little landmines in this DNA that can kill the bacteria that gets stuck there. The neutrophil dies in this process. There's no coming back once you spew the DNA out. But it's a very, very, very efficient way to respond to an infection. And so this is my little, picture here of a neutrophil. And again, the goal of this net is to capture and destroy bacteria. The really cool thing about nets is there's actually a similar mechanism in plants. So there's actually a connection between human immunology and even plants, which I think is just astounding. So I'm happy to talk a little bit more about that if you're interested later. So we have these nets. They're very unique to neutrophils. But neutrophils also really are good at phagocytosis, just like macrophages. So they're really good at eating and destroying bacteria as well. So neutrophils are really important. I also have a video of a neutrophil. And unfortunately, we really can't capture neutrophils doing nets. I wish we had really good, let's see if it works. I wish we had really good videos of a neutrophil actually spewing its DNA. But what I can show you is how incredibly determined neutrophils are to capture bacteria. So what we have here, let's see if I can get a little bit bigger. Might not get bigger. We'll just leave it. So what we have here is this is a neutrophil, okay? And right in front of it, if you see these 2 little dots, those are bacteria. Now, many bacteria are capable of movement. So they actually will be chased by immune cells, and in particular, neutrophils. So all these other little things you see here, these are red blood cells. So we have this white blood cell, a neutrophil in this case. We've got some bacteria. And then everything else you see that kind of just stays steady, those are going to be red blood cells that this neutrophil is going to move around. So if we watch this, do you see how this little bacteria is moving? Yeah, and that's a neutrophil chasing the bacteria, and it does not give up. And it's just moving those red blood cells away, and then eventually it does catch that bacteria, and you'll see that too. So it's almost there. Oh, it caught it. And then you can actually see that bacteria dissolve. It's going to be destroyed through phagocytosis. So it's pretty spectacular. So in addition to, doing this, it's also capable of spewing these nets in addition to really catch these bacteria and contain them. So neutrophils are really specialized and very cool, I think. I mean, I'm super biased, obviously, because I just love the immune system, but I think the immune system does contain some of the just absolute coolest cells in the body. All right, so let's talk a little bit about neutrophils and aging. So again, neutrophils are part of the innate immune system, and we just still need a lot of research into what's happening with neutrophils and aging. So again, I'll share with you a little bit of what we know. So again, our major functions of the neutrophil are these crazy nets, as well as they're very good at chasing and eating bacteria. And so there are some research articles out there that show that just like macrophages, neutrophils are a little bit less able to eat and destroy bacteria. There are also some papers out there that show they have a reduced capacity to make these nets, okay? So ultimately, that would lead us to believe that with aging, neutrophils are a little less functional and maybe contribute then to overall increased susceptibility to infection, right? It is not so straightforward with neutrophils. I also can find a lot of research out there that show in certain situations with age, you actually have neutrophils that are creating these nets when they shouldn't be. And that can be actually a bad thing because these nets are very destructive. So what this can cause is increased inflammation when they should not be there. It can cause destruction of our own cells that should not be destroyed. And so with aging, these particular hallmarks of nets and neutrophils have been associated with something that we like to call immunopathology. What immunopathology means, you see the pathology there, right? So pathology means some type of damage. Immunopathology means damage caused by your own immune system. And I can tell you that particularly with severe COVID, what we are seeing in those patients in the lungs are neutrophils that are just making these nets when they're not supposed to, and that's causing a lot of the damage we see in the lungs with severe COVID. So, and that happens a lot more in aged people. So it's interesting. This is not straightforward. It can depend on the situation. It can depend on the person. But we definitely see cases of both decreased function or too much function with neutrophils depending on the situation. So obviously a lot more research needs to be done with these cells as well. I forgot how much time I have. Is it like an hour? Oh, okay. We got time. I just start talking and don't pay attention. Okay. So I just wait till my students start putting their stuff in their bags. I'm like, oh, okay, it must be time. They get sick of it. All right, so where we're at now, we've talked about some of the very quick responses of the immune system. And those responses are extremely important. And we know even less about them than we do about what I'm going to talk about next. And so let's imagine we've got this little, infection going on. The pathogens are increasing, and we have our innate immune response responding, right? Well, what if the innate response isn't good enough? Sometimes it is. But many times, the innate immune response, containing macrophages and neutrophils, as well as some other cells I haven't talked about, can't take care of the infection by itself. So what needs to happen? We got to call in those big guns I referred to, okay? So we need an even more increased, specialized immune response to really deal with this infection in your body. And that is now where we kind of morph from an innate immune response to what we call an adaptive immune response. So two major arms of the immune response. Innate, fast, first to respond. Adaptive, takes more time to develop, but is a potent, potent weapon. So what we're going to talk about is the 2 cell types that make up your adaptive immune system. So we'll talk about what they do normally and how they're impacted by age. So imagine, we've got this infection happening, it's sending out those danger signals, right? And so what you're hoping is you get an innate response first, but then what happens is the innate immune system can actually activate those big guns, and that includes the B cells, okay? So B cells are part of the adaptive immune response, so they're activated a little bit later on in the immune response. And what these B cells do, and this is an actual picture of a B cell, is they're an antibody factory. That is their only goal. They have one job, and that is to make antibodies. And so in this picture, I just think these antibody factory B cells are so cool because they basically switch their entire cellular purpose to make antibodies. And the way that cells make antibodies or any type of protein is they have to have their DNA wide open so that they can use the genes and the DNA to make protein. And then they have lots of what we call endoplasmic reticulum, which is used to actually make the protein. So DNA gives you the instructions for making the protein. And then in the endoplasmic reticulum, that's where the protein is actually made from those instructions. And so what you can actually see in a B cell is evidence that the B cell's only purpose is to make proteins, which includes antibodies. So in this picture, this B cell here, so the B cell is this entire thing right here. This is its nucleus, and it is huge. You can take my word for it. This nucleus is where the DNA is, the instructions. And so this nucleus has to be huge because its DNA is just wide open, getting ready to give the instructions that are needed for making antibodies. And then I don't know if you can see these little ribbon-like structures that are around the nucleus here. That's endoplasmic reticulum. So anywhere in the cell that's not full of this nucleus is just full of the other part we need to make lots and lots of antibodies called the endoplasmic reticulum. So you can actually just see in microscopy, you can differentiate the B cells that are making antibodies versus the ones that are not, which I think is pretty cool. So B cells make antibodies. So let's talk a little bit about why we would want antibodies, right? Why is our antibodies so powerful? Antibodies have a lot of functions. So remember, B cells are making antibodies. So what that means is a B cell makes these little antibodies and then secretes them, meaning it releases them outside of the cell, and that's where they do their job, okay? And what antibodies are spectacular at is binding, so attaching to bacteria and viruses and just covering them with antibodies so they can't do anything, okay? So it basically just immobilizes bacteria and viruses and prevents them from doing any damage. So antibodies are really very powerful in the immune response. And so what we know about B cells and antibodies with age is a bit more than what we know about some of the other cell types I've already covered. But it's interesting because there's a little bit of conflicting data with B cells as well. So here's a B cell. Remember that its whole goal is to produce antibodies, which are very powerful in helping to contain infection. And first of all, what you should know is that B cells come from the bone marrow. B cells, bone marrow, right? Actually, interesting fact, B cells were actually originally discovered in birds where they're made in the bursa. So birds have bursa instead of bone marrow. It's like a weird little organ. I don't know, I don't study birds. But that's how they were originally named, B cells for bursa. We just got real lucky because they're also made in the bone marrow in humans, so we didn't have to like awkwardly rename them in humans. It still works out. So B cells are made in the bone marrow, and once they're made in the bone marrow, inside of your bones, they're released into the blood, where they become part of your white blood cells in the blood. And in aging, there is evidence that in older people, they make less B cells in the bone marrow. So you get less B cells making their way out into the white blood cells, which can have, obviously impact responses to infection. This is still debated in the field about whether or not this is happening to an extent that makes a difference. Because in humans, you still make a significant amount of B cells, even with age. And so it's debated how much of an impact that actually makes in an aging immune response. So I think future studies kind of have to be done. I can tell you that most immunologists use mice as a model of the immune system. And we can do a lot more with mice, obviously, than we can with humans. And in mice with age, there is no reduction in B cells. So it's tough because we can't actually use our major model for studying the immune system with this particular aspect. So it's a little tough. So there is some evidence, though, that there's reduced production. And there is evidence that even with the B cells that you get with aging, they have less production of antibodies, and those antibodies are a little bit less capable of doing their job. So you might see, less antibodies being able to bind, and they might not be able to bind quite as well to these things. So overall, maybe slightly less function of antibodies as well. Now, of course, we would think that this particular side of things would probably result in a decreased immune response to an infection. Not so clear, though, because, again, in certain individuals with age, we actually see more antibodies that are actually doing things they're not supposed to with age. So if you've heard of the term autoimmunity, what that means is that your immune system starts attacking you. That is not supposed to happen. So the examples of autoimmunity would be lupus, where you have antibodies that actually start attacking your own cells and killing them, causing all kinds of awful problems. Rheumatoid arthritis is where you get antibodies that attack proteins in your joints when they're not supposed to even be there, causing all the problems associated with rheumatoid arthritis. Those are just some examples. And we know that cases of autoimmunity increase with age. And in a lot of people that we see that in is because they actually have increased antibody production and function. Unfortunately, that function is targeted against healthy human cells instead of something like a bacteria. So it's a problem. Again, this is something that we need to be, able to better understand. How do we, try to increase B cell function in the right situation without potentially accidentally making everyone autoimmune? It's a problem. So there needs to be a lot more basic research to understand what's happening here in order to be able to, I think, do something about it. All right. So we talked about B cells as part of the adaptive immune response, later to respond, but very powerful. The other cell type is my absolute favorite, and that is the T cell. So again, we have danger signals being sent out by pathogens. Those have to work through the innate immune system, in particular for T cells. But ultimately, these danger signals activate innate immune cells. I have an example here in green, which then will activate the adaptive cell, the T cells. So T cell stands for thymus cell. So T cells come from the thymus. And T cells, there are two main types of T cells. One of them is a helper T cell, and one of them is an assassin. So the helper T cells, they're like the conductor of the orchestra response. They have their little cellular hands. They don't really have hands, but just picture them having hands. They really have their hand in every aspect. They guide the immune response to be the most efficient against whatever you're infected with. They're extremely important. So in HIV/AIDS, the virus actually kills these helper T cells. And because when HIV progresses to AIDS, there's basically very few or even no of these helper T cells, that's why the entire immune response completely disappears. That's why people with HIV/AIDS are susceptible to all kinds of infections and eventually will die from an infection. Because you remove those, the orchestra conductor, the orchestra disassembles. So the other cell type that's also a T cell are the assassin T cells. As you can probably guess by the name I've given them, they are spectacular killers. So we'll talk a little bit about what they do in just a second. So T cells, extremely important. This is also where I have spent the majority of my time in research. I study the assassin cell type. So I'm very partial to them. So let's talk about T cells and aging. This is really where my dissertation has been. I'm really going to focus on the assassin T cells here. That's where if you read my dissertation, that's what it's all about. I don't suggest reading my dissertation. I'll just kind of give you a little rundown. So I've got T cells, and as a reminder, T cells come from the thymus. The thymus is just like this very goopy gland, I guess, as I would say, that's situated right above the heart, okay? And when you're first born, the thymus is huge. It is just massive. And then after birth, even to early childhood, it starts to decrease in size. So it's the biggest when you're born, and then it decreases with size all throughout life. In fact, the whole reason why T cells were even discovered is because, decades ago when babies needed heart surgery, right, to repair like a hole in the heart or whatever, even during routine surgeries, the thymus is so big that it covers the heart. So doctors just removed it. Get out of the way. We don't know what the thing does. Not good, right? So basically you remove the entire source of T cells, and those children all died early on of infection. So we knew then that the thymus must have something important going on for immune responses, and there is how it was birthed that T cells were discovered. So now when surgeries happen, same surgeries, right? They just remove a portion of the thymus and leave it mostly intact, and that's way better. So I mentioned that the thymus decreases with age, right? Even in kids, by the time you reach puberty or young adulthood, it's much smaller than it was. We know that it continues to decrease into older age as well. And so we give that a special term as T-cell immunologists. We call that thymus involution. Really, what that means, again, fancy term, it means the thymus is getting smaller, and with age, it actually starts to turn to fat. So the thymus not only is getting smaller, it's losing its capacity as a thymus. It's actually becoming very fatty. It's very interesting. So we know because of this thymus involution that we get much reduced T cell production. So this is definitely happening. This is not like kind of wishy-washy like the B cells. You have a lot less T cells being pumped into your bloodstream with age. What that means is you have less T cells to respond to new infections. Okay? So that's a problem. Not only do we know we have less T cells with age, we also know that their function is downregulated. So what I'm showing here is an assassin T cell. And its major function is to patrol your body for cells that might be infected. So it's really good at virus infections. Do you remember how I told you that a virus needs your own cells, right? And so what happens in a viral infection is the virus gets inside of your cell, takes over that cell, basically makes it like a little incubator for making more virus. The problem with viral infections is it's hiding out in your own cells. So your immune system wants to ignore it, right? Usually your immune system is really good at knowing this is dangerous. Oh, that's me. I leave that alone. I fight that. Viruses hide in you. Luckily, you've got these assassin T cells. They're really good at figuring out which of your cells actually are harboring or hiding virus infections. So what they do, these assassin T cells, is they recognize a potential viral infected cell, and they basically bombard, like bomb, that cell with all these really toxic things, causing that cell to die. So if you cause the cell to die, you also can get rid of the viral infection. So that's what these assassin T cells are doing. Interestingly, we know that T cells are really good at fighting cancer too. So T cells are also capable of seeking and destroying precancerous cells. So that's why my lab studies T cells in cancer, because we think that if we understand that better, we can use T cells as a weapon against cancer. So this is supposed to be happening very efficiently. What my dissertation shows is that this does not happen as efficiently with age. These assassin T cells just are not as good killers. Unfortunately, what that means is you get a reduced response to infection, right? These things build on each other. So my whole dissertation is really about the why of this. And I won't go into detail, but we know that there are certain things that happen specifically in an old T cell versus a young T cell that we might actually be able to fix. So it's kind of an interesting continuation off of my dissertation project that now other students in that lab are continuing. All right. So we've kind of built the building blocks now. And now what I want to do is apply them a little bit, okay? So I mentioned that kind of the final goal of the immune response is to make this thing called immune memory. And so let's talk about what that actually is, and then we'll talk about, how that's impacted with age as well. So you've seen this before, right? We have pathogens that are trying to increase their numbers and, cause this infection. And then you have an innate immune response and adaptive immune response that's responding. Ultimately, what you want is that immune response to actually take out the infection, right? So if your immune response is functioning correctly, you'll be able to get rid of that pathogen, get it out of your body. Now, remember that other goal of the immune response turning off, right? So you don't want to keep this massive immune response hanging around if you don't need it anymore, right? So this is, we immunologists, we call this as contraction. So most of the cells that were responding, killing that infection, they'll actually die. It's a good thing. You don't want them around anymore because they could probably cause some problems. You want them to work when they're needed and go away when the job is done. The nice thing is they don't all go away. So some of them stick around. And these are the cells that we would call immune memory. The cells that can do this are only the adaptive cells, B cells and T cells. They can stick around long term. And so let's say you happen to get reinfected. That happens, right? So let's say you get exposed to that same bacteria, that same virus. Now you're not starting from scratch anymore. You've got this protective memory hanging around. What this means is you get this massive, fast response. I mean, it's quick. It takes care of that pathogen so quickly, hopefully you don't even know you're sick, or if you do get sick, it's very mild. Then you have another period of contraction, and actually if I were to follow this out further, it would leave this memory kind of long-term at an even higher level. So if you got reinfected again for the third time, it would be even faster and more efficient. This is the whole purpose of creating memory. This is the whole purpose of vaccination. So what we do with vaccination is instead of making this first exposure a real pathogen, we give you a vaccine instead. What we're hoping to do is then create this immune memory that hangs around, and then if you happen to get infected with the actual bad thing, your immune system doesn't know the difference. It's just going to respond. That's the whole point. That's also why sometimes we have booster vaccinations, because imagine if we gave a vaccine here, got this nice memory population, we give another vaccine, you get an even increased immune response and even more memory cells then that could potentially respond to the real thing. Okay? So that is the goal with either making immune memory to a real infection or making immune memory to a vaccination. It's just using the immune system for exactly what it was built to do. So some infections and vaccinations are really good at making a very stable immune memory populations. So we know, like, for instance, with smallpox vaccination, that immune memory will last your entire life. It's probably the most efficient vaccine. Other vaccinations, like the flu vaccination, for instance, is just not quite as efficient. So it just depends on the type of vaccination and the type of infection. I mean, I would say in terms of immunology, probably at least half of the immunologists in the world have in the back of the mind, how is what I research going to impact making better vaccines? So, there you go. So let's talk about what happens in the same scheme with aging now, okay? So I've told you already that there are some things that happen with the innate immune response and the adaptive immune response that impact just the immune response in general. So what does that mean in terms of memory? What does that mean? So in this graph, we have what you would, kind of expect to see in terms of an immune response for maybe a healthy young adult. Now, first of all, with an immune response in an aged individual, we just start with less, right? We probably have less B cells and less T cells for sure, so that can kind of cause an issue. Let's say we get exposed or vaccinated here. That overall response is decreased. It also takes a bit longer. It contracts. Hopefully you're still getting rid of whatever you're infected with, or you're still getting a response to vaccination, either or. But then there's a problem. We actually see that the number of memory cells usually is lower in aged people. So what that means is let's say you get re-exposed, re-vaccinated, whatever, you get less of that kind of robust secondary response. So ultimately, the immune response is just less. And that extends to that protective memory, okay? So this is kind of the overall schematic here. And I don't want to leave you a complete downer, okay? So what we've noticed until recently is that most vaccines that are used in the United States are less efficacious with age. You may know that already. This is why sometimes we try to give separately designed vaccines. So for instance, there's a flu vaccination that's recommended for older people that tends to be more efficacious, makes a little bit more memory, tries to help that out. However, really the vaccines that we've had in the United States especially, they're just really lacking in the ability to create a really protective response in everyone. All right, so our conclusions this far are, and I don't have too much more, so we'll have plenty of time for questions. We know, I hope you'll agree with me, that the immune system is definitely profoundly impacted by age in many ways. This ultimately, generally speaking, leads to reduced responses to infection as well as vaccination. And so this is something that we really need to study. And so what I want to talk about now briefly, because everyone always asks, so I thought I would just bring it up anyway, is how about our current problem, right? What do we know about immune responses to SARS-CoV-2, which is the virus that causes COVID-19? And what do we know so far about how aging impacts this, which is not very much right now. So what I can tell you, and you may have seen these graphs, I pulled this morning from the CDC. So this is the most up-to-date graph. What this graph is showing is death that has been associated with COVID. And so what it's showing is all deaths in the United States as of today, and then it breaks it down by age group, right? So as you can see very clearly from this graph is that people aged 65 years and older especially are making up the majority of deaths due to COVID. Also, definitely more prevalence of severe infection. So just like what we know for many infections, elderly people are more susceptible. That's not new. I'd be very surprised if COVID had a different graph than this, okay? So what we know about SARS-CoV-2, and I know this is really small, so I'm just gonna kind of point out to you what's important here to understand. we know now a lot about how this virus infects you. So this virus, what it does is it's got these little proteins that stick out from it. It's called a spike protein. You may have heard of this before. And that spike protein is the reason why the virus can attach to you. So what it does is that spike protein will attach to cells, particularly in your upper respiratory tract or in the lungs. So upper respiratory tract is more like your nose, kind of all the stuff up here that's involved with respiration. Your lower respiratory tract, we mean the lungs. So this virus can go in both places. It can attach to your own cells, and then remember how I told you viruses need to get in, right? So the virus gets into your cell and makes your cell a little virus incubator, okay? Now what we know is that your immune system, especially when you're young, is actually really good at responding to this virus. So you make B cells that make antibodies, and you make T cells that can destroy viral infected cells. And in many people, this is enough. Takes care of the infection, no problem. Obviously for other people, doesn't happen so efficiently, right? And so, as you know, there was a push to create an effective vaccine against this particular virus. And I am ecstatic because for the first time in a very long time, we finally introduced some more ways to give vaccinations, okay? And these methods have been in development for decades, but finally got the funding to actually go into clinical trials. So 3 types of vaccines have been approved worldwide for the fight against COVID-19, and they all work with the same basic mechanism, okay? So there are protein-based vaccines, which are not approved here yet in the States, probably won't be because the other 2 work so well. And then we also have what's called a viral vector vaccine, as well as an mRNA vaccine. So let me tell you what they are, okay? So I'll orient you to what's happening here. This is the virus, and do you see these little sticky outy parts here? That's the spike protein. What we know is if we can make an immune response that can basically inactivate the spike protein, the virus won't be able to get into your cells, take care of the virus, okay? So all of the vaccinations in the United States target that spike protein. We're trying to make an immune response against the spike protein to basically not allow the virus to infect. And so that the way that this is done, the Johnson & Johnson vaccine is a viral vector vaccine. So what they do is they take a teeny piece of virus that only would make that spike protein, nothing else, and they put it in a little package, and then they use that as a vaccination. So what happens is your own body produces that spike protein for a very short amount of time, but that's just enough to rev up your immune response. So you remember how I showed you the immune response where you get like a response, it goes down, and that makes protective memory? That's exactly what you're doing here. You're just using that little protein, you're making a little response, and then hopefully getting protective memory so that if you get infected with the real thing, it's either no infection or a less severe infection, okay? So that's the Johnson & Johnson vaccine. The mRNA vaccine, which is the Pfizer and the Moderna vaccines, they basically do the same thing, just a slightly different way. So what they do is they use a different way of delivering that spike protein to your body. Your body makes that spike protein, honestly, for probably like less than an hour. After you get vaccinated, it all happens within like that first hour, okay? And then you get an immune response, make this protective memory, and that includes both B cells and antibodies, as well as T cells. So that's what's happening here, is we're just taking advantage of how we know that immune responses happen normally against viruses like SARS-CoV-2, and then having this vaccine technology to take advantage of it. So this is where I want to go now and leave you hopefully on a high note, okay? So I've told you that vaccinations in general just have not been super effective with the elderly. It's a huge problem. Now we've got these two brand new technologies. It's still creating an immune response, right? That's not changing. It's just how we deliver the vaccine that's changed here. It's ridiculously effective. It's amazing. So for the first time ever, we have a vaccine that in the clinical trials protected people between 65 and 72 years old just as much as everyone else. And if you read articles about this, you see words like this: unprecedented, unexpected, and success. So the immunology community, and at this conference I just got back from last night, everyone's so excited about this because maybe finally we have a way of vaccinating that will actually be effective. The cool thing about the technology that's used in this SARS-CoV-2 vaccination is you can swap it out. We could vaccinate against flu the same way. And so there's this huge push now to use what seems to be a much more effective way of vaccination in other diseases too. So I'm really excited about this, and I'm excited to see where the field goes with this. All right, so conclusions overall. Some of them are the same we've gone through, right? Aging is profoundly impacting the immune system, but we really need to know more. We need to understand the mechanisms. We need to know what's the situation where immunity goes down, but then in other people, the immune system causes autoimmunity. What's the change there? Why is that happening? And all is not lost. So this, first of all, is a major focus of research, both in the bench component as well as the bedside component. And also the recent successes, particularly with the SARS-CoV-2 vaccination, has really opened up a lot of questions, a lot of funding, and a lot of people who are interested in studying in this. I can also tell you that it's not just vaccination that's being impacted. One of the huge areas of research right now is to see if we can, it's called rejuvenation of the thymus. Can we make the thymus less fatty and more productive to make more T cells with age? And there are some drugs that can do that. So there's actually clinical trials right now to see if that's a way to improve overall immunity with age too. And that's just, a couple examples. All right, so I do want to acknowledge a program called BioRender. It's a free program that I use to make a lot of my figures, both in the classroom as well as for talks like these. They just have really outstanding graphics that are free to use. And so I wanted to acknowledge them. And then we've got some time for discussion and questions. So I'd be happy to take anything you would like to talk about.
Thank you so much. I'm going to go ahead and present a question that I have in the chat. I just have one this morning. Why do babies produce such large numbers of T cells? Do they have that much infection or inflammation?
I'm not sure if a certain source is being referenced for that, but actually babies don't produce a lot of T cells right away. So when a baby is born, it's born with a very ready-to-go innate immune system, but it actually takes a couple years for the adaptive immune system, including T cells, to fully develop. So, the reason why the thymus is so big, right after birth is because we want to start filling that kind of empty compartment with T cells as soon as possible so that when the baby is exposed, to those infections, it's ready to go. I don't know if that helps clarify. I'm happy to do a follow-up with that too.
Okay, Susan, we've got several in the classroom.
What can we do to maintain our own robust immune system, either through lifestyle or supplements and things like that?
So the question was, what can people do, in lifestyle, et cetera, to really maintain as robust of an immune system as you can as we age? And, to be honest, there are varying opinions about that. So overall, healthy lifestyle choices very much impact immunity throughout life. So stress, exhaustion, malnutrition, all can reduce immune responses, which then with age can just be compounded. there is very little to no evidence that taking any type of supplements or drinking green tea every day or something, that is not backed by peer-reviewed science. That's just mostly money-making grabs, to be honest. So just making those healthy choices, trying to stay active, especially later on, like in life, especially like over the age of 80. We have this thing that's been recently termed, it's called frailty, which now has a very scientific definition. And that means that we find that people who lose the ability to become active very rapidly become frail, and meaning that they just are more susceptible to damage when they fall. You know, it has a lot of impacts, but it seems to really impact the immune system too. So yeah, you know, if I find a magic cure or something, I'll let you know. But right now, it's a natural part of a bodily process. So being as healthy as possible is really probably the best thing I could say.
I do have a couple more questions in the chat, so I'll take a turn here. I actually have about 6. Let me go back to the beginning. How do genetics impact immunology?
A lot. Yeah, I mean, I could give a whole course on that. So we know that it's the nature versus nurture debate that is debated in everything, right? So we know for sure that certain genes impact your immune system. So for example, some people we call it a genetic predisposition, where they'll have certain genes that make their immune responses just at higher risk of developing things like allergies, right? So we know there's definitely a genetic component to that. But we also know that the environment plays a big role, and that's actually what my lab studies. The environment plays a big role too. So it's not necessarily going to be like, if you have a certain gene, you're definitely going to get asthma. But these things work together. So yeah, I mean, if you have a specific question about a certain situation, but the answer is absolutely your genes very much impact immunity.
Thank you. Why did you specifically mention that US vaccines are less effective in the aged? Is there another approach elsewhere?
I would say that up until recently, the United States has had a hard time advancing vaccination technology. Whereas in certain other European countries, as well as some Asian countries, that hasn't been so much of a barrier. A lot of that has to do with how we get funding for research in the United States. And so I can't think of a specific example off the top of my head. I just would say that I personally, in my opinion, feel like this technology that we're using for SARS-CoV-2 vaccination could have been implemented a long time ago. Do you want to take a couple questions from in the room? I know we've got some hands up, so maybe we can switch back and forth to a couple from the room, a couple from Zoom.
Anybody in the room?
Looking at you extending your conversation about the RNA,
Mm-hmm.
if it's been around for all those years, how come it wasn't used? And, okay, so, and part of the discounting factor about don't want the vaccine because it's so new, well, you're inferring it's not new.
Right. So the question has to do with, if the technology for both actually the viral vector as well as mRNA vaccines have been around so long, why has it taken this long? And, there is a part of the hesitancy to get the vaccine is people are worried that A, it's such a new technology, like, is it safe? And B, it's been quote unquote rushed, right? The mRNA vaccine technology was proposed over 20 years ago and has been successfully used in animal models for decades. And so it's been around for a really long time, but there was never a push to get it in the clinic, mostly because we couldn't get enough funding for it. So vaccines, in order to become vaccines, they start with basic science. They move on to animals, and then you have to get significant funding to push for a clinical trial. Clinical trials happen in stages. You get phase 1, where it's more of a safety, making sure it's safe in humans. Phase 2 is to figure out, okay, if it's safe, let's figure out what we would need to do to make it the most efficacious. Do we need 1 booster? Do we need 2 boosters? What do we need to do? And then the third is where you then release the vaccine to a much wider audience using what you've learned from the previous 2, and then tweak it if needed before full FDA approval. Each stage of those clinical trials takes millions upon millions of dollars. Where do you get that money? Pharmaceutical companies usually. So in order to get that type of money, it's not coming from the government. You actually have to convince pharmaceutical companies to, convince pharmaceutical companies that they'll make money if they support the clinical trials, and that's very difficult, especially when we already had all these vaccination options out there, even though they weren't great in certain populations, right? So what happened with the SARS-CoV-2 vaccination is the government, before clinical trials even started, fully guaranteed the funding for all 3 clinical trials. So it was simply the funding. No safety measures were taken out of commission. Full clinical trials happened, but instead of having to have years between each stage of the clinical trial while you wait to try to convince people to give you money, the funding was guaranteed from the get-go. That's the difference. That's the only difference.
Okay, I've had 2 Pfizer vaccines and the booster.
Yeah.
In the future, am I probably going to need another booster?
So the question is, so things are changing, right? So the question is, right now, the 3rd booster for mRNA vaccines especially has been now recommended for all adults. And that's because what we've noticed is that after 6 months post the 2nd vaccine, you do start to see a slight decrease in especially your memory B cells. And so if we give you another boost, you get another response, and then you get that population back, right? And so it is very possible that we're going to see something similar like what we see with the flu vaccine, where just to keep those memory cells healthy and active and fully protective, you might need to get multiple boosters. But to be honest, we don't know yet. We need to wait and see. Now that that 3rd booster has been approved, we'll have to, there are so many researchers and clinicians that are following that very closely now to see what's going to happen. So it's possible, but we don't know yet.
I'm going to go ahead and ask one from the chat. Given the recent spike in cases in Ottawa County, what do you think the case trend will be in January and February?
I'm definitely not an epidemiologist, right? This is not my specialty. I don't actually have time to follow all of the trends, and I don't have the background to be able to make accurate predictions like that. What I can tell you is that, in my opinion, because there's been hesitancy to get this vaccination, we have not reached the critical mass of vaccination, which is why we're seeing the variants. And unfortunately, with the variants, we've found that the vaccine is still very effective against the variants, with the current variants. But what the fear is that the more we allow this virus to percolate because people aren't getting vaccinated, the more chances that another variant's going to come out where the vaccine won't be quite as effective. So it's hard to predict, but it is certainly possible that we could continue to see cases going up. Right now, the Delta variant accounts for all cases. So there was the Alpha variant, Beta variant, and then one other little variant in there that didn't really do much, and now it's the Delta variant. So are we going to get like the Epsilon variant? I don't know. I hope not. But convince all your friends to get vaccinated, and maybe we have a better chance.
Our bodies are continuously producing B cells and T cells, as you explained. Where did the macrophages and the neutrophils originally come from?
Yeah, good question. So the question is, I talked about where B cells and T cells come from. So what about other immune cells? So every immune cell originates in the bone marrow, and that actually includes T cells as well. So we call that hematopoiesis. Hematopoiesis, again, fancy term, that's hard to say, even harder to spell, believe me. That is the process of making blood cells. So hematopoiesis starts in the bone marrow where you have stem cells that then differentiate through a process to become these different types of cells. So that includes your red blood cells too. So your red blood cells are made in the bone marrow, and then you get cells that are still kind of stem-like, and then they go to the thymus to become T cells. If they stay in the bone marrow, they'll become B cells. And then you also have cells in the bone marrow that become macrophages and neutrophils and the other immune cells. It all starts in the bone marrow. Yeah.
I have a
Another-
couple more in the chat. Let me hit one here real quickly. Are there any way to improve white blood cell counts to do a better job when born with very low counts? In parentheses, to the point of needing shots.
I want to make sure I understand that question.
Karen, do you want to unmute yourself and ask that? Or I can,
Sure,
okay.
I can do that. So, when I was born, I had like very, very, very few white blood cells.
Sure.
So they had to start giving me these, I think they call them gamma globulin shots or something.
Mhm.
So I had to go in for routine shots to
Mhm.
try to develop a white blood cell count because I was, sick all the time.
Mhm.
And I'm just wondering, as I age, facing things like these COVID viruses and stuff, and while I can get the vaccine, I tend to have a lower, what do you say, efficacy
Mhm.
or
Yeah.
response to it. So is there something that I can do that will improve my army of white blood cells?
The short answer is probably not.
Okay.
So from what you're describing, again, I am not an MD, nor have I been involved with your case. I always
Yeah,
feel like I
yeah.
have to disclaimer all of this, right?
Yep.
From what you're describing, it sounds like you may have had or still have a relatively common genetic disorder that impacts your ability to make especially B cells, but more importantly, antibodies.
Mm.
So in those types of genetic disorders, especially, the best treatment is just to replace those antibodies that you don't make. So if you've ever heard of the antibody infusion treatment for severe COVID, like if you have really severe symptoms, you can just get approved now to head over to Spectrum, then you get a shot and you feel a lot better. What they're doing is they are injecting antibodies in you. That's it. They purify antibodies either from donors or they can make them in the lab, and they give you more of them to boost your response. So when you're talking about your gamma globulin, globulin is another name for it's another name for antibody. So when you were a baby, they were giving you more antibodies just through IV to protect you, while hopefully your B cells caught up.
Got it.
Yeah.
Okay.
So
Thanks.
you want to ask a question? Sure.
Yeah, just to piggyback on that, now we've got pills or some kind of treatment for people that actually have
Mhm.
COVID. And how does that treatment affect the disease or the infection? And I think it's the same thing now with cancer patients that are not doing some kind of infusion.
Hmm.
How does that treatment stuff work with regard to the cells?
Sure. Again, that could be an entire course, honestly. So in particular, I think you're probably referring to the recent approval of the FDA for the Merck treatment, which is a pill. And that is actually an antiviral pill. So if I go back to, I'm very visual. So what that antiviral pill does is it basically stops this part of the virus. So those antiviral pills can stop the virus from using your host cell as an incubator. So it's a drug that prevents viral replication. It's a viral replication inhibitor. So it's not immune-based. It's actually attacking the actual virus's mechanism. And it's actually, antiviral medications are used all the time. The treatment used in HIV is antiviral medications, multiple antiviral medications used all at once that target the virus to prevent it from doing what it does. So that's the way that medication works. It's kind of cool. It seems to be pretty effective if used pretty early on in infection. And then in terms of cancer, I think all I can say right now, and maybe you'll have to invite me back because I do enjoy talking about cancer immune responses and then cancer immunotherapy. So in my opinion, the field will move to be using what we call cancer immunotherapy a lot more to fight cancer, because we know that the immune system normally is very good at preventing cancer. So when cancer develops, something must go wrong with the immune system. That's why cancer is more prevalent with age too, because the immune system function goes down. As a side effect of that, more cancer. And so what if we can figure out a way to make better immune responses to cancer via drugs, via infusions, giving more cells, et cetera, et cetera, et cetera, to make your already what should be a potent weapon against cancer potent again. So there is a lot of clinical trials and now approved therapies that we would lump under that immunotherapy that are very interesting for cancer. And in fact, that was probably the other major topic at the conference I just came back from was, now that we understand more of the basics, how do we really apply that to improving health using the immune system? So yeah, that's what I'll say for now. You can invite me back. I'll talk about it more.
Which we would love to do that. It is 11, and so I just want to invite anyone who has a prior engagement, if they need to leave, they're invited to do so. But there's another question in the chat, if you'd be willing to answer that.
Yeah, I can stick around probably for another 20 minutes before I have to go lecture at Grand Valley. So I'd like to eat before that.
Yeah, as far as the Zoom classroom, we'll just stay open for a few more minutes, and then I will have to close that. And then people in the classroom, if they want to walk up and thank you for being gracious with your time. There's 2 questions. I think you answered her third one, but I'll just ask both of them and you can answer them in order. Are there tests that can be given to see if boosters are individually needed? So I'm assuming they mean for an individual person. And then how long did it take for the polio vaccine to make it from research into arms?
Individual tests, kinda. So you may have heard of antibody tests for COVID. So what they're doing there is they're not testing to see if you have virus. They're testing to see if you've got antibodies in your system that are either A, present, or B, actually capable of doing their job. So there's 2 different types of antibody tests, just to look for amount, and then to look for do they actually work. So, I mean, technically speaking, yes, we could do antibody tests on people. If they still have a very high amount and super functional antibodies, then the booster may not be needed at that time. And this is actually done for other vaccinations. I don't know if you've ever had a hepatitis B titer done, maybe. Like every time I, do something new, I have to get my blood taken and just a quick test for a hepatitis B titer. That's exactly what they're doing. They're testing to see how many antibodies you have left from your childhood hepatitis B vaccine. Do they still work? If they happen to notice, they're kind of getting on the lower side, they might say, hey, we want you to get another hepatitis B vaccine just to make sure you're protected. So I see some people nodding in here, so I can tell that, that's a little bit more familiar. So we could apply that same technology to COVID if necessary. At this point in the game, I would highly recommend that if you can get a booster, you just get a booster. Even if you already have still some functional antibodies left in you, you're only going to benefit your immune system to get that booster. And then the other question, oh gosh, I already forgot. Someone remind me.
The polio vaccine.
Oh yeah, I don't know. I don't know how long the polio vaccine, and there's 2 types of polio vaccines. There's the oral vaccine and the injected vaccine. The oral vaccine has been discontinued in the United States, but it's still used in a lot of different countries. I can tell you on average that before COVID, vaccinations took minimum 10 years to go from idea, research in a lab, to FDA approval. And it all had to do with funding. So I'm guessing that the polio vaccine was somewhere in there too.
I have a question related to cancer cells.
Okay?
You mentioned earlier that T cells can recognize precancerous cells.
Yep.
So what has changed on the surface of the cells that they can become targets if they're not yet cancerous?
Mm-hmm.
What has changed in on the membrane or other characteristics?
Yeah, good question. So that's also a huge field of study. The question is, how does the immune system potentially recognize cells that might be becoming cancerous and nip them in the bud before they can actually cause cancer? And so a hallmark of cancer is mutations, right? So the reason why a normal cell in your body becomes cancerous is it accumulates, we estimate, between 4 to 7 mutations. And those mutations work together to cause cancer. So if you define cancer, it means abnormal and invasive cell proliferation, meaning that your cells are just expanding when they shouldn't be and nothing's controlling it, okay? And so those mutations that happen in your DNA cause that. So when those mutations happen, they can also cause changes in parts of the cell that the immune system would see. And so if that change happens, it can actually look like it's not yourself anymore. So the immune system thinks, wait a second, does that cell have a virus infection in it? That's not recognized as me anymore. Something's going on. Is this danger? And so that's where you can actually get an immune response. We call those neoantigens, Neo-antigens. That doesn't happen in every cell cancer type, but that's one mechanism of how surface of your own cell will change enough that your immune system is so sensitive that it can recognize even those slight changes. Another weird thing that happens is a lot of cancer cells start expressing genes and stuff that is only normally expressed like in embryonic development. So like example, let's say you have a liver tumor. Sometimes the cells of the liver will start turning on genes that you would only find in the brain. Your immune system also knows that's not normal. So that's another way that precancerous cells can be detected and killed before it causes a huge problem. There must be other mechanisms out there too. Still field of research. You're welcome.
I think that that's all we have in the chat. So
Okay.
what I'm going to do is leave, if you, first of all, we need to thank you. And yes. Do you want to go ahead and close the class in there by taking the mic?
I'm sorry, say again?
I can go ahead and close the class. We just thank you so much.
Oh, you're
On behalf of
welcome. That's my
everybody at
pleasure.
home and everyone in the classroom. It was just a wealth of information, and we appreciate you sharing your gifts
Yes.
with us. And we do hope that we see you again. Thank you very much.
Thank you. You're welcome.
Well done.
Thank you.
I forgot to mention.